So if we look globally at the different types of signaling cascades, for this short piece, we're going to focus on these signals here. So generally, these are hormones or steroid hormones. And for us, this will be cortisol. And these steroid hormones, what's unique about them is they don't bind a membrane-bound receptor. Rather, the receptors for steroid hormones sits either within the cytosol or within the nucleus. The other thing to note about steroid hormones is they signal primarily by modulating gene transcription. So if we look at our flow of information here, we can see that going from gene to primary transcript is our level of transcription. So again, steroid hormones will really influence whether transcription of a gene product is increased or decreased. And in these cases, if transcription is increased, what we'll see is an increase in a protein product. And that increase in protein product will ultimately increase flux through that pathway. And this will be the case with cortisol when we look at its signaling. So generally speaking, there are two major classes of intracellular receptors. You can see here that if this is our hormone, it will move into the cell through the plasma membrane into the cytoplasm. In this first case, our hormone can bind a nuclear-bound receptor. So for this first class, we see that the receptor is already within the nucleus and bound to a response element. These response elements are DNA sequences that are unique and that the protein will bind to. These are upstream of genes that will be regulating. So our example here is thyroid hormone, which its receptor is bound to DNA within the nucleus and functions as a transcriptional repressor in the absence of hormone. You can see here that when hormone is present, the repressor is displaced, and binding of the hormone leads to transcription of the downstream gene product. For us, we're going to talk about cortisol as our example of a steroid hormone, and its signaling is shown here. So again, our steroid hormone cortisol will diffuse through the plasma membrane, and in this case, cortisol binds an intracellular receptor. If you'll notice, this intracellular receptor is in the cytoplasm bound to a heat shock protein. Upon ligand binding, you notice a conformational change that typically exposes a nuclear localization sequence signal, and the protein is then trafficked to the nucleus, where it will bind to its response element, denoted here as a glucocorticoid response element, which again is a sequence on the DNA strand. Once bound, this will then increase transcription of the downstream gene product. So again, there's two types of intracellular receptors to be aware of. One is DNA bound in the absence of ligand, such as this case here with our example of thyroid hormone signaling. And for us, more classically, we'll talk about cortisol signaling, where its receptor is present in the cytoplasm, bound to an adapter protein or heat shock protein as illustrated here. And when cortisol is present, will cause a conformational change and allow the bound receptor to then translocate into the nucleus and influence gene transcription. So let's finish with a quick question. So elevated levels of cortisol we know can increase blood glucose levels. And one possible mechanism for this is increased transcription of genes involved in which of the following pathways. So we know from previous discussions that cortisol is going to be elevated in the fasted state primarily. So we're really asking the question is which one of these pathways is present and active in fasted state metabolism? So we know in the fasted state, these are going to be primarily catabolic pathways, so we won't be having synthesis of either fatty acids or protein. So that leaves us with glycolysis and gluconeogenesis. So these two pathways are opposing, and in the fasted state, we're primarily producing glucose through the process of gluconeogenesis. So our correct answer here is B, gluconeogenesis. And what we'll see in subsequent weeks is that cortisol does in fact increase the transcription of genes involved in gluconeogenesis. Specifically, it will increase transcription of the gene product from PEP carboxykinase, which is involved in this pathway.